Today we're going to be talking about amino acids and proteins and about transcription, and because we're talking about transcription, we'll also be talking a little bit about translation. But first, let's just go over the announcements. So we have a worksheet due today. Please hand it in before you leave. We have a lab due on Friday. There was a question about whether or not the lab included the trait wheel at the very end of the lab in the manual. You don't have to do that. You've done plenty with the rest of the lab. And on Thursday, we're going to have a quiz on today's material in class. And it's going to be a general, kind of an overall quiz, oversight, kind of a broad concepts type quiz. So on that note, I did not grade the last quizzes, but I do have your makeup quizzes from quiz 3, where I gave you the opportunity to do edits on them. So I did grade those. It's in also your grade book on Blackboard, so that should all be adjusted already, those quiz 3 grades. Mm-hmm.
Did you want the pre-lab questions on it too?
Yes, you could do the pre-lab questions as well for the lab. And again, as usual, you could either type them up or take a picture and upload that. Either way is fine. Lexi?
Quiz 4.
So quiz 4 was the online quiz where you had to order things. So I went through the quiz 4s, and actually I have to be a little bit easier on the computer program because they were pretty difficult to grade. So for now, I've downgraded them to a homework. So they're not a quiz weight anymore, and I've reduced the weight of them overall quite a bit. So they're only worth 5 points. Your normal homeworks are worth 10 to 15 points. So that's what I decided to do with those, because I know that there were some problems with them. And it wasn't what I expected from my side, and I don't think that it was what you guys expected from your side. So I figured this was a way to kind of mediate that. Claire?
You did well on it. Is there any way that you can grade more?
So it still helps your homework grade a lot. Yeah. Good question though. Let's see. I think that's all the logistics stuff. I wanted to start out because some of the material we're talking about today is a little bit dry. We're going to be talking about chemistry again and molecules, but this stuff is really important in the big picture because we're talking about amino acids, and they are what make up proteins. And the shape of a protein determines its function, and proteins determine basically what a cell is doing at the moment. And I wanted to show this image, so I'm sure many of you have heard about this before, that sickle cell disease is caused by the change of a single amino acid in one protein. So hemoglobin, obviously an important protein. Hemoglobin is the protein that actually binds to oxygen in our blood and delivers that oxygen to our tissues. So it's necessary essentially to bring oxygen to your whole body. And when you change one single amino acid, then this hemoglobin protein is going to misfold. And the bigger picture of that is that when that protein misfolds, then it actually affects the shape of the entire cell, and the protein no longer works, so it's no longer carrying oxygen to your tissues. And worse, then these cells can start to clump and block your various pathways throughout your body where blood is flowing. So even the normal shaped blood cells that have hemoglobin that's working could then be prevented from getting to your tissues to deliver oxygen. And again, this is just the substitution of one particular amino acid. If you changed other amino acids, you wouldn't have the same effect, but again, this is only the change of one amino acid. It has a huge impact on the overall individual. So we'll talk more about this towards the end of class, but I did just want to bring this up as an example. So actually, I'm going to ask everybody to go to Kahoot right now, and we'll have just a mini quiz. So this is not going to count towards anything. This is just as a review and to see what background information all of you are coming in with. So we're doing classic. And here's our PIN number. Feels so strange to play without the sound. But I don't know how to put the sound up here. Oh, there we go. I didn't know how to put the sound up. 13. All right. So, let's begin. And again, this is stuff that some of it we went over in class, some of it you may have read about, some of it you may or may not know from previous classes. And it's okay if you don't get these right. So which of the following processes results in the copying of DNA sequence to make new DNA? And we did do this one in class. We did this early on in the semester. I'm just trying to move this so that we could all see it easily. And the answer here is the copying of DNA occurs during S phase, and that is called DNA replication. And it happens, remember, before mitosis. S phase technically does not occur during mitosis, but as many of you remembered, the copying of DNA is necessary in order for mitosis to go forward. All right, next question here. Anthony is up at the top. Nice job, Anthony. What two processes are necessary for gene expression? Great, so we did really well. We haven't talked about this in a while. We did mention in the beginning of the semester, transcription and translation are two processes that lead to gene expression. And we've talked about some of these other things. Crossing over, again, is for meiosis, and then transcription is for gene expression, and then hydrolysis is a type of reaction, chemical reaction that we've talked about a little bit. And photosynthesis is for capturing light energy, and we'll talk about that in a couple of weeks. Okay, Nicole. Great job, Nicole. So what molecule is read in transcription, and what is made? Remember that transcription is not the same as DNA replication. So DNA is read and RNA is made. So I apologize, I should have put read instead of copied. But DNA is the molecule that is read. It is the the transcript that is read, and then RNA is made. The red one here, DNA is copied, DNA is made, that would be DNA replication. And then this yellow one doesn't actually really occur, but there is this connection between RNA and amino acid sequence, which we'll talk about in a minute. And if you look at the really big picture, we do have all of our genes coded for in our DNA, and eventually they end up as amino acid sequences, but that process doesn't happen directly. Okay, so next, Nicole, great job. Anthony, keep it up. What is the molecule that is read in translation, and what is made? Great. So many of you caught on that this was the second part of the last question that I didn't really explain. RNA is used, so we make RNA during transcription, and then after that in translation, we read that RNA and make amino acid sequence from it. And then DNA being copied and RNA being made, that's transcription. And let's go on to the next question. There's only a couple more. Oh, Riley's up at the top, but Claire's on fire, so I don't know what that means, but good job, everybody. What is the enzyme that does the bulk of the copying during DNA synthesis? So DNA replication, what's that enzyme that we talked about? What protein? So it's DNA polymerase 3, but if you chose DNA polymerase 1, that's okay. That's pretty close. DNA polymerase 1 is actually the polymerase that fills in the gaps when the RNA primase, the primer, is removed. But these DNA polymerase 1 and 3, very, very similar molecules. It's just that DNA polymerase 3 is a lot faster, so that does most of the DNA copying, but I'm glad that everyone remembered that DNA polymerase is the thing that we're looking for, so great job. And then topoisomerase, does anybody remember what that is? We didn't focus on it too much, but it was in the reading. No? So that was a molecule that cuts the DNA and lets it unwind so that the DNA doesn't get too tangled as helicase opens at the replication fork. And the ribosome is used in translation, which we'll talk about in a little bit. Okay, Riley, the top, great job. What is the enzyme responsible for catalyzing reactions, or basically performing the reactions in transcription? So remember, transcription, reading DNA, making RNA. So the answer to this one was RNA polymerase. Similar to DNA polymerase, it copies a DNA sequence and then makes a chain of nucleotides. Helicase opens up the DNA helix. So interestingly, RNA polymerase is very similar to helicase as well, and it can open up that helix. So any of you who chose that, there is a connection there. And then DNA polymerase, nobody chose. We just said that makes DNA. And ribozymes, that was a great guess because it has the same kind of root of that word. But I'll give you a hint. The ribosome is a type of ribozyme, so that might help you in a couple minutes. Okay, oh, Caitlin's now at the top. Let's see what happens now. What is the enzyme responsible for catalyzing reaction in translation? we're going from RNA to amino acid sequence now, translation. Great, the ribosome. So I'm glad to see that a lot of you are familiar with the ribosome. Again, reads RNA and then uses the information in that RNA to make amino acid sequence or kind of nascent peptides or proteins, that is. And then telomerase helps to copy the ends of chromosomes, and proteinase K is so does anybody know what proteinase is? What does ACE mean? Does anyone remember the ending of ACE? This is a type of enzyme that they use to break apart other molecules. So a proteinase destroys proteins. DNA ACE or DNase destroys DNA. RNase destroys RNA. So one of the things, oh, here's a good connection. When we were using meat tenderizer in the lab and other chemicals to extract just the DNA, we were trying to destroy some of the proteins, right? So that would be, have some proteinase activity, the meat tenderizer. And soap has some proteinase activity too, it's just not great at it. That's mostly for lipids. Okay, Caitlin up at the top, Riley close behind. 2 more questions. What are the bonds between nucleotides in DNA and RNA? Don't get tricked. So phosphodiester bonds, because we have very similar molecules in DNA and RNA, if you recall. We only differ by what chemical group on the sugar?
Second, carboxyl.
The 2' OH group, yeah. And so we have those same phosphodiester bonds. And then, so phosphodiester bonds, most of you knew that was the right choice. Peptide bonds are in what molecule? Between amino acids, right, yeah. I know, we wanted to talk about proteins, but remember, phosphodiester bonds for all ribonucleic acids. Okay, Nicole's on fire now. Where's the fire extinguisher? So, last question. What are the bonds between monomers of a protein? So what is a protein made up of? And then what are the bonds between those? Great, yeah. So proteins are made up of amino acids. There's no such thing as an amino bond. I know that was tricky. And the bonds between amino acids are peptide bonds. Great, excellent. Thank you, everybody. Nicole up top, Claire and Kate. I'm sorry, Anthony, but you did great for most of it. So,
Is there a way to find the answers, like, on these answers?
yeah, so I could open up the quiz so that you guys could play it by yourselves. I just need to figure out how.
Okay.
But I will make a note to do that.
Try writing.
All right. So on the note of the last question, I just wanted to start by going over the connections again between DNA, mRNA, polypeptides, and proteins. And I want to really quickly go over some of the terminology. So we go from DNA, or reading DNA, and creating mRNA. This is transcription. Then, going from mRNA, reading that messenger RNA, we read that, use information to make an amino acid sequence. That is translation. And when we have the amino acid sequence, we tend to think of that as, oh, everything's finished, like that's the protein, but that's not true, because we have, this is just basically a type of information, a type of code, right? We have an amino acid sequence, but proteins are 3D. So we also have to have folding. And that's when we make a functional protein. And again, that's really important because if you just have a peptide chain that is not folded, perhaps it's in the wrong environment, so it's the wrong pH or things like that, then it won't necessarily be functional. Or if you heat up a folded protein, then it becomes unfolded, and again, it's not functional. So we have this really strong connection between shape and function. So I also wanted to mention that you're going to see some different terminology, which I emailed everyone about yesterday. So polypeptides are chains of amino acids and proteins are folded chains of amino acids. Sometimes we also refer to unfolded proteins or unfinished proteins as nascent proteins. Like they're being formed. And then one other thing I wanted to point out was that amino acids are sometimes referred to as residues. So you might refer to a single amino acid as a residue. I don't know where that came from. It doesn't seem to be really connected to anything else, but you will definitely see this. And again, I think that we saw this question earlier in the semester. Do both of these types of cells, bone cells and muscle tissue, contain the same DNA?
Yes.
Yes, they do. But how do they differ? They differ in the genes they're expressing, so they have different RNA and they have different proteins. Great. So it's really important. Okay, and this was some of the vocabulary that I have there. And this is just a reminder of the connection between chemistry and biology. So we're going to be talking about this a lot again. So just remember that the properties of a molecule depend on what atoms are in that molecule. What charges do they have? How stable are they? What's their hydrophobicity or hydrophilic tendencies? So just keep this in mind. And we're going to go back to, if you recall, this chart of important chemical groups to know, because we're going to talk about amino acids and their composition. So one of the groups we talked about was an amino group. So of course amino acids have the amino group, that is the NH2 group on the amino acid. And then we also talked about the carboxyl group, and that is another important group on an amino acid. So every single amino acid has an amino group and a carboxyl group. And what else have we talked about previously? We have this central carbon as well. So this is a different depiction of an amino acid, and then there's a third depiction there. Again, we have the amino group, carboxyl group, and they're both connected to the same carbon. This is the central carbon. Then the other chemical groups are the hydrogen, which every single amino acid has that one hydrogen, so that this carbon has 4 bonds, so that it's stable. And then we also have an R group. Does anybody remember what an R group is?
Like what identifies the
It's what gives the individual amino acids their different identities. So an R group in general, if you're a chemist or a fan of chemistry, means that you could put a bunch of different chemical groups here, but the rest of them would stay the same. And so these are all related molecules because all 3 of these groups would be the same, but then you have different things here at the R group. So for example, if we were going to talk about DNA and RNA, which maybe we're a little bit more familiar with than amino acids at this point,